are going to get started, and I want to welcome you all to the Great Lakes Phragmites 2024 webinar series. So we counted, and this is actually, I think, our 54th webinar that we've hosted as a collaborative. So we're getting up there in the numbers. And I'm Teresa, and I'm joined by my colleagues online, Sam Tank and Nicole Angel. And we are hosting today's webinar on behalf of the Great Lakes Phragmites Collaborative. And our presenters here today, Wes Bickford and Kaira Ligget, are from USGS's Great Lakes Science Center here in Ann Arbor, Michigan. And they will be presenting multi-year cut to drowned management of Phragmites populations can control flooded patches. And we're going to give folks a few minutes to kind of flow into the webinar room. So while we do that, I'm just going to give a few housekeeping slides. For those who've been to webinars before, sorry, I feel like this is the same spiel I always give, but for folks who are new and haven't attended one of our webinars, this is just a quick overview of the Great Lakes Phragmites Collaborative. So the Great Lakes Phragmites Collaborative is a partnership to link people, information, and action on invasive Phragmites. And the collaborative is coordinated jointly by the Great Lakes Commission and then USGS Great Lakes Science Center, and with funding from the Great Lakes Restoration Initiative. And our collaborative facilitates communication among all stakeholders across the region and assists with development of management tools and programs, and serves as a resource center for information on Phragmites biology, management, and research. And the collaborative is led by the Multi-Agency Advisory Committee and applies principles of collective impact to advance a common agenda for Phragmites across the region. So that is a little bit of what we do. And just for something fun to look at, here's our common agenda. And the Great Lakes Phragmites works towards advancing member strategies that are outlined here in our common agenda. So this helps us clearly define what is the intention of the collaborative, develop the shared understanding of the gaps in the problem area surrounding Phragmites, and highlight areas and strategies that we are working on or wish to work on. And if you want to learn more about our regional efforts and what we're actually doing in the moment, you can visit our website, which I believe I have on the next slide. Yep. And then a big component of what we do as a collaborative is we run a program called the Phragmites Adaptive Management Framework, or PAMF as the acronym is called. Oops. And what PAMF does is it utilizes participatory science to provide data-driven Phragmites management guidance through an adaptive management framework. Basically, it means that we have established a way to learn what Phragmites managers are doing out in the field and turn that data into annual guidance to help managers make effective and efficient management decisions. And to learn more about our PAMF program, you can go to that URL on the screen, or you're welcome to email pamf@glc.org, which will get you in contact with our PAMF coordinator. Okay, that's fine. I'm missing a slide. And then other things that the Great Lakes Phragmites Collaborative does is we host just a bunch of different resources for anyone who's managing Phragmites. So we encourage you to check them out by going to our website, which URL is on the screen. We have recordings of past webinars, all 54 of them. You can join our listserv and connect with other Phragmites managers. You can sign up for our newsletter, which has recent Phragmites news, as well as a quarterly research roundup of published literature in the Phragmites field. So a bunch of cool stuff and resources that we encourage you to check out. It's a really great website. Again, there is our URL, and then if you have any questions about what the Phragmites Collaborative does or want more information, you're welcome to email us. And then, of course, follow us on social media, because we love having new followers at GLPrag. And also today's webinar will be recorded and will be posted up on our website. So you all should get an email from me saying when it's posted, but it'll be posted on this Great Lakes Phragmites Net website. For anyone who missed it, or if you want to share it, it will be there. And lastly, before we get to the presentation, just some basic housekeeping. So as you came into this webinar, you should have all been automatically muted with your cameras off. And to avoid any disturbance, we ask you to keep it that way. So if sometime during the webinar your camera pops on or your mic pops on, we ask you politely to turn those off. And it's just the camera and microphone button here at the top. You can just click it on or click it off, excuse me. Also, we have two different ways to communicate. We have a chat bubble, which is right here in the green, all the way to the left. And like Sam put in the chat, if you're having any tech issues, if you can't hear us, if something's going awry, please let us know. And we'll respond to you back in the chat and try to fix whatever technical issues we're having. We also ask that we'll have a Q&A portion at the end of our webinar for our presenters. And we ask you to put any questions to the Q&A portion in this little Q&A bubble right here. You don't have to wait to the end of the presentation. If a question comes to you, 10 minutes in, please add it. We'll just wait till the end to ask the question. If you post a question and the presenters don't get to it before you leave, the webinar is recorded, so you're welcome to view the recording and see them answer your question for you. So just again, chat bubble here all the way to the left for tech issues, and Sam, Nicole, or I will respond to you. The Q&A bubble is questions for our presenters at the end of the Q&A portion. Okay, my little spiel is over. So now it's my pleasure to introduce today's presenters. So Wes Bickford is a biologist at the USGS Great Lakes Science Center in Ann Arbor, Michigan. Wes has over 10 years of experience researching Phragmites control options, and he has a background in wetland ecology, invasion ecology, and microbial ecology. And Kaira Ligget is a student contractor at USGS in Ann Arbor, Michigan, and is currently researching cut-to-draw management strategy on Phragmites, as well as a conservation project concerning wild rice. She has received her bachelor degree from University of Michigan in ecology and evolutionary biology, and will be returning in the fall to work on her master's degree. So thank you today for talking with us, and I will let you guys now take it away.
All right. Thank you for the introduction, Teresa. Appreciate it. And thank you all for being here today. I'm excited to be here again to present the results of our research that we've been working on for the past several years. Teresa mentioned this is the 54th Phragmites Collaborative webinar. I believe we presented on the 50th one last year. So if you were able to catch that, we hope to provide some updates of our research since that time. If you didn't catch that one, you can go back and look at it where we were a year ago, but we should be able to cover some of that today. I would like to just go ahead and recognize our funders, the U.S. Geological Survey and the U.S. Fish and Wildlife Service, for funding this work. And I'm excited to present our results today. All right, so let's get this going. So we are gathered here together because we all have a mutual interest or maybe disdain for this plant. We're all interested in finding new management strategies or making our management strategies work better for Phragmites because we know all the impacts that it has across the region. One thing that I want to point out is, I like to help everyone understand how water levels have influenced Phragmites spread throughout the region. And we all are pretty aware that water levels are kind of on their way down after being really high. We're coming off of a high water period. But before that high water period, there was a really long extended period of low water. And it really tracks well with the expansion of Phragmites in the region during that same time. And I'll give you an example. in the Saginaw Bay. So this is a site near the Saginaw Bay, near the Fish Point State Game Area. And at this site, if you look at the aerial photograph from 1999, this was before water levels reached their recent minimum and stayed there for a long time. So water levels were still fairly high. And you'll notice in that water, you don't see a lot of vegetation growing. It's pretty much open water there. In the aerial photograph in 2011, this is after water levels have been down for at least 10 years. They've been really, really low. And so Phragmites was able to take advantage of that exposed lake bed and just expand, create this kind of wall of Phragmites that was really pretty characteristic of the Saginaw Bay area and other shallow water areas of the Great Lakes, like in Western Lake Erie, in the Green Bay in Wisconsin. Areas like that where Phragmites could take advantage of that exposed lake bed and create these dense monocultures. So we all know then that water levels went up, and you'll notice in the aerial photograph from 2018, this is before they reached their maximum, but were pretty high for a while. The Phragmites in the north part of that is stressed. It's breaking up, it's becoming really patchy, and the stuff in the south had been eradicated. So managers and landowners were able to take advantage of that stress on the plants and used, I think it was a chemical herbicide to remove Phragmites from that area, but really took advantage of that stress. So two important things to take away from this. One, Phragmites can persist in high water. And two, under that stress of high water, it's a really good opportunity to manage it because it's kind of knocking it with two things while it's already stressed. So how does the plant survive in high water? All plants need to breathe. All living things need to breathe, and plants are just the same. Phragmites is the same. So it takes in air through its leaves, through the stomata, breathes in through the green parts of the plant, and actually takes the air down through the rhizomes, and it breathes out of the dead culms. So it's kind of like breathing in the green and out the brown. And so when water levels are even higher, it's able to snorkel, right? So it can as long as a portion of that plant remains above the water level, it can still breathe. So it's still breathing in the green and out the brown. However, if you cut off that flow of oxygen, if you cut the plant below the water line, or let's say if it breaks, naturally via wave action or something like that, we've disrupted the flow of air and oxygen, and the plant can't survive that. It dies. So this is the idea behind this cut to drown management strategy, is that we're just cutting the plants below the water so they can't breathe. They're drowning. And so importantly, this is not new. We didn't create this. This is something that's been used in the region for a long time. And especially in areas like in Canada, where there's restrictions on certain herbicides that you can use over water. So the managers there have gotten really creative with different cutting tools and ways to control Phragmites over water and have had a lot of really good results from that as well. And not just hand tools that they've been able to use. There's cool tools that can do this on a large scale. This is a amphibious vehicle called a truck saw, which is like an awesome name for Phragmites control tool that can cut underwater in a larger scale setting. So if you want to hear more about this work, I'd recommend you go back about 2 years ago, listen to a talk that Dr. Janice Gilbert gave on the Phragmites Collaborative, and she talks about a lot of these strategies that her group has been using to control Phragmites underwater. So despite its kind of widespread usage, there was a lot that we didn't quite understand about this management strategy. And so we developed a few questions about the strategy. One, what's the optimal timing for cutting? Should we be cutting early in the season? Should we be cutting later in the season? What's going to be most effective? And two, are the rhizomes actually inviable after they're drowned? So if the plant isn't resprouting, does that mean it's actually dead? It's kind of actually hard to tell if Phragmites is dead. You may have noticed that in the field. It's just kind of weird. And then also, what if the clone extends into low water? So if the plant is not fully submerged, if the stand is not fully submerged, some of that plant may be unflooded, and can it actually rescue the flooded portions if you cut it underwater? So these are the questions that we set out to answer. And we were successful in getting funding from the Science Support Partnership Program through the Fish and Wildlife Service. And we proposed doing a greenhouse study and a multi-year field study. So we're going to present results of those today. So I mentioned that last year we talked about this work, so I'm not going to go into great detail about our greenhouse experiment, but I'll say this was led by myself and Spencer Whidden, and we put together a greenhouse experiment at the Botanical Gardens here at the University of Michigan, and we're able to manipulate different water levels to cut at different times and to see how the plants survived. So you could see we had different cutting treatments, an early cut and a late cut. We had different submergence treatments, a partial submergence where the plant was partially submerged in water, but when cut was fully submerged, and then a total submerged plant, which is something that was completely covered in water. So these plants were propagated from rhizomes we collected from the field, established in small pots, placed into these cattle tanks at different depths using bricks to vary the depth, and then flooded with water, allowed to grow for 120 days, after which we harvested them. And we were interested in looking at the below ground biomass, so how much root and rhizome mass was created by the plant, and then the carbohydrate concentration in that plant. And that gives us a sense for kind of like what we call in this presentation the growth capital, or its ability to regrow. How much does it have in reserves to be able to make a new shoot? Then with some of those plants, we actually overwintered them and regrew them the following year to see if the rhizomes were viable. What we found was that cut to drown was really effective in these small plants. We were able to kill these plants that were in these small pots by cutting them underwater. We also found that the cut timing didn't really matter, that as long as they were cut to and drowned, they were killed from a early cut or from a late cut. And importantly, cutting alone was not effective at killing the plants. And they needed to be drowned as well as cut. But total submergence, if you could submerge the plant completely, was effective at killing the plant. So we put together a manuscript and were able to get this published last year. It came out in Wetlands Ecology and Management. If you're interested in learning more about this study, I'd encourage you to check out the manuscript. But you'll notice, okay, these plants were tiny. Yeah, we could flood these plants, we can kill these plants, that's great. We learned some things. But you know, it kind of lacked realism, right? It's like, we know that it may be a lot easier to kill a potted Phragmites plant than it is going to be to kill an entire stand. And so we wanted to know how well this translates to a field environment. So we took these ideas and these things that we learned to a field setting. And so I'm going to hand it off now to Kaira Ligget, who's going to explain everything that we have learned from our field study over the last couple of years. Kaira played an integral role in helping to develop some of these field strategies and compiling the data and analyzing the data. And so she's going to take it from here. So Kaira, I will hand it off to you.
All right, thank you, Wes. Yeah, so as Wes said, my name is Kaira Ligget, and yeah, I'll start off by talking about our multi-year field experiment. Next slide. So for our field experiment, we were able to obtain 2 field sites pictured here. Next slide. And so over those 2 years, we were able to maintain these sites. Our first site was Metzger Marsh, located in Oregon, Ohio, within the Ottawa National Wildlife Refuge. And this site is a very open water, very large site, and it borders Lake Erie with very deep water as well. Moving forward, we'll just be calling this site our coastal wetland. Our second site, Garner Road Marsh, which we'll be referring to as the impounded wetland, is located in Saginaw Bay, or near Saginaw Bay, and contained within the Fish Point State Wildlife Area and managed by the DNR. So this site was or is man-made and controlled, its water level is controlled by a pump. And so this site, differing from our coastal wetland, is more contained and also it's not open water, so it's smaller as well. So for both of these sites, we allocated our plots and we subjected them to treatments varying in cut timing as well as cut frequency. Next slide. And so let's start off by talking about cut timing. So it's important to note that timing is very significant in the Phragmites life cycle. So depending on what point in the season it is, it corresponds to the movement of energy within the stand. And so we'll get into the specifics of what I mean when I say energy, but let's just talk about the phases. So starting with dormant, it's during the colder seasons, the energy is stored and being contained within the underground rhizome network. During the growing phase, as it gets warmer, germination occurs and vegetation starts to grow, you see the energy movement happening upwards. And so the energy is going into creating those new stems. And then there's flowering and seeding that takes place and shedding of the seeds. That is the translocating phase. And during that phase, the energies starting to migrate back down and into the rhizome network, and it sort of starts over here. So essentially, our cut timings align with different periods within this phase, and we wanted to understand how different timings may be affecting or not affecting the cycle. And so in addition to cut timing, we also had cut frequency. So I forgot to mention on the first slide that we had our early cut, we had a late cut and a continuous cut, but we'll be focusing today on the late and the continuous cutting that we did over our 2 years. And so our late cut, we did one time, and our continuous cut after the first initial cut, we came back and cut every 2 to 3 weeks. So in year 1, we had our single late cut and our multiple continuous cuts that happened. And then in year 2, we took the plots from both of those treatments and subjected all of them to continuous cutting in year 2. Next slide. So during the field season, year 1, we applied our cut treatments varying in timing and frequency, and then we returned every 2 to 3 weeks and we monitored stem regrowth as well as applying our cuts to the applicable plots, recutting. And then at the very end of the season, we collected rhizomes from all of the plots, and I will talk about in a little bit why we did that. Next slide. One thing that we thought was really important to mention, especially from a management perspective, is the amount of time and energy and the number of people it required to remove the biomass from our coastal wetland site. Because that site was more open water, we needed to remove that biomass so the Phragmites wouldn't float to other areas within the marsh. And so you see the photo in the center, that is from one patch of Phragmites, the initial cutting of one patch. And so over this season, in the first year, we had to do that for, I believe, roughly 12 plots. And so that gives you an idea of just how much work that was. So in year 1, it required a lot of time and energy. Next slide. And so field season year 2, we proceeded at the same. We maintained our plots from year 1. We applied our treatments and then returned to monitor every 2 to 3 weeks, monitoring for regrowth, recutting applicable plots. And again, we did our end of season rhizome collection. Next slide. And so something really interesting that was happening at both sites was that it was really evident what what treatment plots received in year 1. So we see some photos here that were from our coastal site. You can see the photo on the left compared to the photo on the right is the stem density, it's much more sparse. And so the photo on the left is of a plot that received continuous cutting in year 1, while the plot on the left, or excuse me, on the right, only received 1 cut in year 1. So this was during field season year 2, and is really evident that, you know, the cutting was having an effect on things. Next slide. Similarly, we observed the same thing with biomass removal. At the coastal wetland, if you remember those large boatloads for one stand, contrast that against these photos here, we were able to collect the biomass and apply our cuts from multiple plots, and, you know, just a little bit of Phragmites here. Everyone was still able to get into the boat. Lots of room. So it was significantly less work in year 2. Next slide. And so now we'll talk about the preliminary results. So before we get into that, it's important to talk about our measures of success, which Wes did touch on a little bit. The first thing that we were looking at was above ground growth. And so the way that we were looking at that was the stem density at each plot, number of stems coming back. The second measure of success was growth capital. And so that was what I was getting at when I was talking about the energy. And so the rhizome carbohydrates is basically how we were understanding how much energy was left. And so for the rhizome carbohydrates, we were looking at 2 different things, percent starch and percent sugar. And so it's important to understand in this case that during the growing season, there's that constant conversion of starch to sugar. So starch is that long-term, and sugar is that readily available energy reserves. And so that conversion is constantly occurring. And so we wanted to understand how our cutting treatments could be affecting those 2 things. And then lastly, we were looking at viability. And the way that we were measuring that was those rhizomes that we were collecting post-season, as I mentioned, we would take those post-treatment and we place them in a growth chamber and assess how many stems were coming back, if any, from our rhizomes that we had collected. And so for our preliminary results in year 1, if you remember, we did find that this method resulted in a significant reduction in all of our measures of success. However, the stands were still alive. And by the way, the cut that was the most effective in year 1 was a continuous cutting. So while the measures of success were reduced, plants were still alive. There's some growth capital remaining. Rhizomes were viable. And so we came to the conclusion at the end of year 1 that this was, in fact, a multi-year process. Next slide. And so now we'll talk about our results from year 2. So we'll start with above ground growth, which again is our stem density. Wes, if you could just click once. We saw that for 2 years of continuous cutting at both the coastal and impounded wetland, it reduced stem density to nearly 0. So these numbers were a lot lower. As I mentioned previously, you could see kind of early on that things were already not coming back as much in those plots. And so at both sites, 2 years of continuous cutting is what was reducing that density. And now we move to growth capital, which again is those energy reserves. Here we're looking at percent sugar. Wes, if you could click once. We see that 2 years of continuous cutting significantly reduced percent sugar. And so it wasn't significantly different from the plots that received one single late cut in year 1 and continuous cutting in year 2. But we do see that those percent sugar levels were significantly depleted, significantly different from the controls. And then lastly, we have growth capital. or excuse me, so almost last, but percent starch, which again is the long-term storage. And so if you remember, I mentioned that there's that constant conversion from long-term, which is the starch, to short-term, which is the sugar. So we see here that 2 years of continuous cutting depleted those reserves, which was really exciting to see. So there's virtually no reserves left for the 2 years of continuous cutting. And this was the case at both the coastal and the impounded wetland sites. And then lastly, we will look at viability, which, just as a reminder, those rhizomes' ability to put up shoots post-treatment. And so for both the, for the coastal wetland, we saw that a single late cut in year 1 and continuous cutting in year 2 reduced viability to almost zero, or to zero, excuse me. And then in the impounded wetland, we saw that 2 years of continuous cutting is what reduced viability to zero. And then in the alternating treatments, you can see this really wide bar, and that's simply due to the fact that there was a lot of variability with resprouting of these rhizomes. But we see that the viability was reduced, which was really great to see. And now I will hand it off to Wes, and he's going to talk to you guys about adventitious roots.
Thanks, Kaira. So I'm returning to a topic that we spent like one slide on last time when we presented, and we showed a slide and we're like, hey, we noticed that one of the plots that had a lot of regrowth also had a lot of these roots in the water, and we're like, I don't know if it matters. And so, just kind of anecdotally that year, we noticed that it looked like the plots that had these adventitious roots were regrowing faster. And so maybe they were helping them grow back quicker from cut to drown. And so one of our interns that summer was Priya Gahir. She was a cooperative summer fellowship program intern with us in the summer of 22, and then started a master's program at the University of Michigan and thought this idea was super interesting and liked working with us, and was like, hey, maybe I could look at this for my master's. And so she's actually presenting her capstone project on Friday this week. So she's wrapping it all up. She's done a really great job at looking at this, and I'm going to completely steal her thunder by presenting some of her data now. So thanks, Priya. So I'll step back and just like make sure everybody understands what adventitious roots are. You probably have seen these in the field if you've pulled up a Phragmites stem that's growing in high water. These are the roots that are growing from the nodes, and they grow above the soil but below the water line. And the mechanism or what they actually do is kind of unclear. We don't know if it's nutrient uptake, if what they're actually there for, but there's been some evidence in other submerged plants that they can provide dissolved oxygen to the plant. So our question was, how are they impacting cut to drown? And it could be significant because, I mentioned before how the plants can snorkel if there's a portion of the plant above the water. And cut to drown basically works by like cutting off the snorkel, right? So you remove that. But if these roots are allowing it, giving it some access to oxygen, it's like you cut off the snorkel, but you give them an oxygen tank. And so these plants can then survive, metabolize, and create new shoots and kind of withstand these cutting treatments. So we wanted to look into this a little closer to try to understand this more. So from a management perspective, she was interested in knowing the impact of adventitious roots on the effectiveness of cut to drown. And so we were looking specifically at how adventitious roots affect plant performance and how that effect depends on dissolved oxygen and the cutting strategy. So Priya set up a really nice experiment in the greenhouse. This is a fully factorial design where we started with plants that were small, much like the greenhouse study I just talked about that Spencer and I put together, where we had small plants, but this time we actually forced them to grow adventitious roots before the study. So we did this by just flooding them, putting them in warm conditions underwater, and they just kind of all created those roots. So they all had some adventitious roots growing on them. And then we subjected them to some cutting treatments. So there was a no cut, a full cut, which was all the way at the soil surface, and then a partial cut. And the partial cut was below the water line, but above the soil surface. So it allowed the plant to have some stem with some nodes and kept some of those adventitious roots present. We had another treatment where we actually removed the adventitious roots. So in the partial cut and the full cut, which had some of their roots still remaining, we just plucked them off. And so those were like continuously removed several times throughout the experiment. So the plant was never able to get the benefits of having them. And then also there was a treatment that added a bubbler. So we were interested in knowing how these adventitious roots impacted the plant in high dissolved oxygen and low dissolved oxygen. So we just aerated some of those pots with like a fish tank bubbler. This is what it looked like in the greenhouse. All the buckets were randomized, but you can see what the aerator creates this oxygenated looking water compared to the stagnant water in the non-bubbler pots, and you can get a sense for like what a partial, a full, and a no cut look like. At the end of the growing season, we counted the number of stems. We also counted the number of stems continually throughout. So how often were they sprouting new stems throughout the course of the study? We looked at total below ground biomass at the end, new rhizome biomass, what portion of that below ground biomass was root, what was rhizome, and then the carbohydrate concentration. And I'm going to focus just on the top two there, the number of stems and the below ground biomass. Okay, so I kept this slide mostly because it gives you a really good picture of what a partial cut looks like. So obviously this is out of the water, but you can see that that stem is cut above the soil line and those adventitious roots are still present. So if that was underwater and those stems were impacting the plant, or those roots were impacting the plant positively, it could be kind of helping it withstand this cut to drown treatment. So after our experiment, we saw that in the below ground biomass, the partial and the full cuts had significantly less below ground biomass than the no cuts. And this was really similar to what we found in the previous greenhouse experiment. Cut to drown works, cut to drown kills or reduces the amount of below ground biomass. And so this was really consistent. We also didn't see any impact of the bubbler or the adventitious root removal on below ground biomass. But where it gets really interesting is when we look at the cumulative number of stems over time. So these vertical lines indicate our two cutting dates over the course of the study. And you'll notice like right away after we did these partial cuts, the partially cut stems started regrowing really fast. And that wasn't the case for the full cuts, and it wasn't the case even for the no cuts. There weren't producing stems at the same rate. And so we ended up with significantly more new stems in those partial cuts than we had in the full or the no cuts. We also saw, if we look at, kind of the what the roots did, in those partial cuts, we saw, the partial cuts had more new stems, like we just saw in the other graph, but we also saw that the roots matter more when dissolved oxygen is low. So when there's no bubbler, the plots with roots had more new stems than the plots without. And so this gives us a sense that, maybe those roots are providing some oxygen to the plant. So the main findings of this were that partial cuts were kind of the least effective way to do cut to drown. And there was no effect of the adventitious roots or dissolved oxygen on below ground biomass or carbohydrates, which I didn't show, but they did regrow a whole lot faster when you did these partial cuts, and specifically when those adventitious roots were present. And so it leads us to believe that the adventitious roots may promote stem growth when dissolved oxygen is low. And so maybe this is what was going on in the field when we saw these plots that just like kept putting up new shoots. Anecdotally, what we were performing in the field was much more like a partial cut. We were unable to get all the way to the surface of the soil at these sites, just due to like how bumpy it was and how it was really difficult to get down really low. And also, we didn't know that was necessary, right? So we were cutting it below the water line. But some of those roots remained, and maybe that was the reason that these were resprouting the way they were. Right, so I'm going to turn it back over to Kaira to explain one other aspect of this research project.
Thanks, Wes. Yeah, I'm gonna talk to you guys really quickly about this. And so Wes, in the beginning, talked to you all about our 3 questions, and so now I'm going to basically explain how we kind of carried out looking into that 3rd question, referring to the partially submerged stands. And so to answer the question relating to partially submerged stands, whether that connection matters when undergoing this treatment, we implemented an additional treatment in addition to cut type connection. So we had a connected treatment and a disconnected treatment, and this was at the impounded wetland site. And so for the connected treatment, we trenched around 2 of the 4 sides of the plots, and for our disconnected treatments, we trenched around all 4 sides of those plots. Next slide. And so let's look at basically like why this matters, what we were doing. So let's say that like the portion to the right of the wooden stake is inside of the plot. So we apply our cut, but you can see that that connection to via the underground rhizome network is still present. Next slide. And so by trenching around the perimeter, we severed those connections, completely cutting off the inside of our allocated plots from the outside, ensuring that there was no sort of transfer occurring or any stems or anything growing from the outside in. Next slide. And so once we trenched our plots, we constructed this quadrat, this 3 meter by 3 meter quadrat, and at each plot, the partially trenched plots, we placed the quadrat down. And what this quadrat did was divide our plots into 4 quadrants, and then we were able to assess in each individual quadrant if there was any sort of pattern occurring as it relates to stem count. Next slide. And so we observed something really interesting. We did see that in the quadrants that were adjacent to those trench sides, there were lower stem counts than in the quadrants that were not adjacent to those trench sides. And so this suggests that maybe connection does may be important, which is an important consideration from a management perspective if the managed population is partially submerged. And connection to the unsubmerged portion, that might be really important. So, and I'll hand it off to Wes to wrap it up for us.
Thanks, Kaira. And I will say we have more data on this, looking at connected and disconnected plots in all of our measures of success. We just didn't have enough time to share it all. But I want to get into kind of the key takeaways from today. So I put together a list of things that I think are really important for everyone to think about. And one is just that cutting works. I think that we've had a good deal of data that suggests that we can reduce the stand density, we can reduce the amount of rhizome carbohydrates or its ability to resprout, and we can reduce viability significantly by cutting underwater. And I'm going to talk about some caveats to all of this, but I think it's really important to realize that this is a really viable management strategy, something that can work very well. Next is that I think that we should maybe think about it as cut to drown and cut to starve. And it's kind of funny. So we were at we all gave presentations, Kaira and I and Priya actually, at a regional meeting for the Ecological Society of America recently. And someone raised their hand after Kaira's presentation and said, you should call it cut to starve. And we were like, okay, yeah, whatever. And then later, I started thinking about it. I'm like, that's actually pretty helpful in a management sense, because if you're thinking about doing this and you're calling it cut to drown, that implies that the drowning is the most important thing. But really, you also need to starve these plants. If they're able to regrow, just getting them underwater is not going to be enough, because if they can keep putting up new shoots, you really need to keep cutting them so they can really starve themselves of all those below ground reserves. And so we have really good evidence of that here. When we look at our percent starch content, and we see that these plants that were in these cut treatments were really starved of their below ground reserves and not able to resprout. So we were able to cut to drown and cut to starve, and that was really, really helpful, and it was the most effective way to treat them. The next thing is that adventitious roots are important. And Priya's research really illustrated that they were especially important in the regrowth of stems. And that could be really important too, because I think what it might suggest is that maybe you cut low, right? Like cutting as low as possible is probably going to be the best way to eliminate those adventitious roots from these plants and keep them from resprouting as quickly. So if you can't cut low, you're probably going to have to come back more often to really starve these plants of their resources. But maybe cutting low allows them to drown before they're completely depleted of resources. The last thing, and Kaira touched on this at the beginning, is that this is super labor intensive, but mostly in year one. So I think that the idea that we have these boatloads of frag and it took us a lot of time and effort to take it away, I think that's really important and something you should definitely keep in mind if you're planning to implement this, but it shouldn't scare you away from doing it. One, because as you scale up, you could use different types of machinery. We were out there with it was just me or one other person with a cutting tool, cutting these stands. That's not the way you'd want to do it for a large population, obviously, and you'd have other tools at your disposal. But the other important thing is that every time you come back, it just gets easier. So, Kaira mentioned that this boatload was from a single plot. It was actually like there were multiple of those, up to 3 from a single plot. So there was a lot from those plots. But in year 2, the pictures at the bottom, that is like multiple plots all in the same boat, and we can all still fit in it. So that gives you a sense for like how much biomass we were getting in year 2, which was a whole lot less. The other important thing about the intensity of how much time it takes to do this is, like, I think we're still trying to figure out what's the optimal timing, because you'll notice that our data suggested that a late cut and a continuous cut was not a whole lot different from continuous cut and then continuous cut in both years. And obviously the amount of effort involved is a lot less for that late cut to a continuous cut rather than doing continuous cuts 2 years in a row. So I think we're still kind of searching for that optimum, like what's the least amount of effort that's necessary to achieve the desired results. And that's something that I'd be interested in talking more about with other people and maybe looking into more. So with that, I will just say thank you very much for having us here. Thanks to the Phragmites Collaborative for hosting the webinar series. I'd like to thank again our funders at the Fish and Wildlife Service and the USGS, and our project partners, the Fish and Wildlife Service, the Michigan DNR, and the Ohio DNR, all of which have been really, really helpful in this whole process, giving us field sites to work in and helping us get set up. And our entire field team. This is a long list of people that have helped on this project. You'll see Teresa's name on this too, because she helped at the very beginning of this work when she was working with us at the Great Lakes Science Center. And also our co-PIs, Kurt Kowalski and Michelle Vanderhaar. So with that, I think we had some time for questions. I'd be happy to answer anything that came in. Thanks.
That was so good, guys. I learned so much. Yes, guys, feel free. You don't have to log off. We have some time for questions. So, yes, like Sam mentioned, please use this Q&A function. That's what I'm going to read from. But yes, thank you. That was amazing and beautiful graphics. I love whoever did all your guys' drawings, and the illustrations of the cutting to drown were amazing and helpful.
Thank you.
Totally Kaira's work.
Kaira, they're so good.
Thank you.
Okay, so as people start with the Q&A, I'm going to read them out, and then whoever wants to answer, feel free. So this is from Alex. Did you guys notice if AR were more adventitious roots were more present in coastal stands versus the impounded stands, or vice versa?
We did notice, so I'd be hesitant to say they're more prevalent in coastal stands because we only had one coastal stand and one impounded stand, but we did notice more of them at the coastal stand. That could be because that site had been flooded for a long period of time, and those plants, were used to being in water and were producing those like pretty much immediately. Whereas the impounded wetland was flooded the year that we started the project. So the plants that were there before did not have adventitious roots, obviously, but we did notice that in year 2, they were starting to develop them, pretty quickly as well. So yeah, there was a difference at the beginning, but as there was more time underwater, those plants, developed those roots just the same.
Yeah, that makes sense. Even though do you guys think it has anything to do with like the dissolved oxygen? I would imagine like this coastal site would be more oxygenated than the impounded wetland, but maybe not.
Yeah, one thing that we didn't get to do was to actually like do a field test of Priya's experiment. We had hoped to do that, but just funding and timing, we didn't have the ability to do it. But we wanted to put out dissolved oxygen sensors in the field to get a sense for if there were trends that we could find between the amount of adventitious roots and the dissolved oxygen concentration of the water.
Future research project for sure.
Yeah.
From Tom, what kind of equipment is available to scale up for larger projects?
That's a great question. One thing I'd say is if you haven't already, go watch Janice Gilbert's presentation. You can scroll down to like a year ago on the Phragmites collaborative webinar page, or 2 years ago actually, and she talks about scaling up. But one of the things that I showed a picture of was that truck saw, and let's see if I can just go back to it. But that thing looks really cool. It's just like a boat that has a huge cutting blade on it. And I think it's kind of like a barge. So like it gives you the ability to like put the cut stems right into that, right onto the barge, the platform of it. So that is something that, when people talk about doing, here it is, the truck saw. When people talk about doing this at scale, they mention, well, I should get a truck saw. So that's something that I think there's like one organization in Michigan that has one, that everybody's been talking about, like how they can, rent it or whatever. And I suppose like for an impounded site, like a Marshmaster could work as well, especially if you're able to like, cut it while it's, just saturated but not totally flooded, and then flood it quickly. You might be able to have pretty good success, and then go back and kind of spot treat after, if it's too flooded for the Marshmaster.
Yeah, unfortunately, they're crazy expensive. We're always like creeping on how much truck saws cost. So I feel like some opportunity to share equipment would definitely probably be helpful to folks.
But they look so cool. Look at that guy.
They're so
awesome.
cool.
I want to be his friend.
Okay, this is from Donald. During what time of the year should continuous cutting, which is the every 2 to 3 weeks, be performed, in your guys' opinion?
Yeah, so we did our continuous cuts. They started in about May, and we went all the way through August. Is that right, Kaira?
Yeah.
Yep. So we did it for pretty much the entire growing season, which was a significant amount of work. But the idea is that we didn't want those plants to have too much time above the surface of the water where they're able to photosynthesize and replenish those carbohydrates into their rhizomes. So, they're using the starches to converting to sugars, using the sugars to make new stems, but they're replenishing those sugars via photosynthesis. So like if we could cut them before they had a chance to really do that too much, we felt like that was appropriate, and that's why we did it about every 2 to 3 weeks during that peak growing season.
From Douglas, if the rhizome system is extending beyond the cut area, will this method still work? So you guys kind of addressed it in the later portion, but
it's a great question. And we did try to, we showed a little bit of the data at the end about that. And we saw that we did see that rhizomes, or that new sprouts were coming up in those areas that were connected. We also saw some evidence of some differences in the amount of stems that were coming up in our connected versus our disconnected plots. I think that if it does extend, the best way to go would be to try to cut off that connection to the unflooded area. So if you have the ability to trench that in some way, we just use shovels, like serrated shovels, to do it, which might make sense for your site, or maybe that's crazy because you had a huge site. But there's probably other ways that you could do it as well. But if you have it where it's gradient that you really can't get around the fact that some of it's unflooded and some of it's flooded, trying to cut off the flow of resources from the unflooded stems is probably the best way to go.
Did you retrench, or was it just like a single, did you worry about the rhizomes regrowing through the trench mark?
We tried to retrench about once a month each field season.
Okay.
Yeah.
That's a lot of work.
Yes.
This is from Grace. What would you say was the biggest setback in year 1 of getting this project started?
Well, I think that we didn't realize how much work it was going to be to cut those, and not just the cutting. The cutting part's easy, it's just swinging a hedge trimmer around, but it's the gathering of all of the stems. And the reason that was important is that they can just resprout in water. I mean, we do this all the time when we're trying to propagate for a greenhouse. We just put the stems in water. And we notice this in the field often. If a plant gets knocked over, it's putting up new shoots from its stems. And so we didn't want to just leave these sitting in water in a warm environment to just create more of a problem. So we had to get them off site. We've done a few little mini projects to see if you make piles with the cut material in dry locations, if they'll resprout, and that seems to work pretty well. They don't resprout from those as much, but unfortunately at the site we just didn't have the ability to do that, and so that meant that we had to put them all on the boat. We had to take the boat back to shore. We had to load them into a truck, and we had to take it to a composting facility, and it ended up just taking forever. So and those were like Kaira's first days on the job, and she was probably ready to quit at that moment. So.
Actually, perfectly leads into the next question is, what would your recommendations be regarding biomass disposal on such a large scale like this? So.
Yeah. I think that so if you have an area that is available that you could pile it up, our small little experiments that we've done suggest that those don't really resprout from the piles, and especially if you were able to tarp them or something for a while so that you can try to reduce any regrowth from those piles. That would be kind of the easiest way, or if you could burn them after they're piled up, if that would work. But without other options, taking them off site is a way that works. It's just hard.
Just a side pitch. There's different ordinances with like how you can dispose depending on where you are with like invasive plant material. So sometimes you can't just take it to the dump. Sometimes you can't compost it. So you always have to like look into like what the area will allow you to do. So
Yeah, really good
just
point.
a little pitch on that. Kind of similar since you guys talked about burning it. This is from John Paul. So with difficulties of the vegetation removal that you guys saw in year 1, what is the possibility of using a dry season burn to remove the past year's vegetation and begin with cutting with the semi-continuous cutting regime, so the less frequent one throughout the year? Do you think this would require less vegetation removal?
Yeah, I do. I think it would still, so if you removed all of the previous year's stems, that would help. At the site at Metzger Marsh, there were some of the previous year's stems present, but it's an area that I think it's decent ice cover and ice shearing and things like that. So it wasn't extensive in my memory. Most of what we had was the previous year's stems. But if you do have a site like that where you've got a lot of dead stems every year, yeah, doing something like that to pre-clear it, I think would help the amount of work that it would take to remove the biomass at the beginning of the year. As to how that affects, the effectiveness of resprouting and all that, I don't know, because you may actually be, giving it more light earlier in the season, which could make it sprout faster or something, extending the growing season. But certainly from like a labor perspective, that could make it a lot easier on you.
There's tons of people still need there's tons of work to be done. There's so many different options. Okay, a fun question. What types of little animals did you guys encounter while out in the field? Did you see any cool stuff?
Kaira, I want to leave that one to you because
Yeah.
If you don't tell it, I'm going to tell one of my favorite stories from the field.
Yes, so yeah, tons of cool wildlife. There's tons of birds, and you'd see frogs, and we saw well, actually, I thought it was a PVC pipe. I was reaching my hand in the water, and I lift what I thought was PVC pipe out of the water, and it was a giant turtle. So
You just grabbed him by the
that was
leg and
great.
like
Yeah, I just grabbed
hoisted him
the
up.
back of his shell, yeah, and I was like, oh my gosh, that was a turtle. Sorry about that.
Yeah, that was my favorite story because I heard a scream and I was like, hey, what? Are you okay? She's like, oh, I just tossed a turtle. Oh, like
That's
that.
amazing. Okay, I'm just scrolling through the Q&A. So the great questions, guys. I think I've addressed all of them.
From Tom, Teresa.
Say that again, Sam.
One more just came in from Tom.
Oh. Why am I not seeing it? You can ask it, Sam, because I don't see where it is.
Yep, no problem. This is from Tom, and it says, so when doing continuous cutting, assume you achieve better results when cutting close to the bottom versus partial cuts, provided you also address the additional roots associated with the adjacent stands.
Yes. Yeah. So, with adjacent stands, is that a question about the connection? I'm not sure about the roots, the adjacent stands. I believe if you are cutting, continuously cutting as low as possible, this is something, again, that we didn't test in the field. Big caveat there. This is just based on our greenhouse study with the adventitious roots. But it seems that these continuous cuts as close to the surface as possible is kind of what would likely have the best results. And if you have a stand next to it, adjacent stand that also has roots, I think it would be way easier to, try to sever that connection than to try to like do anything to, sever those roots or anything like that, because there's just so many of them. So trying to, sever that connection, I think would be the best.
You see any else, Sam? I think mine just has a bit of a lag in the Q&A. Oh, yes. Tom. Can I unmute you?
Unmute Tom. Give us 1 second, Tom, and we'll do that. Okay, Tom, you are able to unmute yourself now.
It was a mistake. I'm sorry.
No problem.
So press the wrong button.
Okay. I think there was
And
one other
then
hand.
Jack, yes, let's
Jack,
see.
you are able to unmute.
Okay, thanks, Sam.
Hello, everyone. The trenching kind of caught my eye.
Yeah.
Here. If you had a vessel that had the ability to trench closer to the shoreline to keep the vegetation from growing back out into the lake, maybe like a trencher that fits on a that they used to put in power lines or underground feeds. So basically, when you cut it at the water line, the roots that are in that area are also being fed from the land or other areas. So how deep do the roots grow?
Real deep. That's a terrible answer, but they do grow pretty deep. We trenched to about it was about a 1.5 feet in depth, and we were able to see results from that. So we were severing some connections, but we didn't know obviously if we severed all of them, because they can grow, a full meter deep, and they can be they go all over the place. I've been in a plot before. We were just trying to harvest rhizomes for growing something in the greenhouse. I'm sort of following this rhizome deeper and deeper, and it just like sometimes they go straight down. It's really odd. So we've pulled them up from at least a meter deep in the past. So it is difficult to know if you've gotten deep enough. We chose about a 1.5 feet. That was about the longest shovel we could find. We did rent a piece of power equipment, a trencher, and took it to that impounded wetland and promptly got it stuck in the wetland. So that's why we went away from using a trencher. But if there was something that was able to be put on, on a boat or something that could go into the water, I could see how that could be really helpful in kind of stopping the flow of resources from those unflooded areas. For sure.
Okay. Any other Q&A questions for them? Now is your time. Okay.
Questions in the Q&A, Teresa.
There's a new one.
No, I do not see any others.
Oh, okay. Thank you. Oh, do you have a follow-up question, Jack, or is your hand just still up? I'm going to assume it's a residual hand. Happens. But yeah, so if you wanted to share this recording, like you wanted to watch it again, your colleague's going to love it. After this meeting, we will upload it to our website. We'll also send you an email with where it's linked at. So you can view all this information again, share it out. The Q&A portion is included in the recording. And then next week, we have another research webinar, same Tuesday time, but same Tuesday, different time, 1 o'clock. So we're doing afternoon. So you can register on our website, the same place you registered for this webinar, and it's going to be super interesting. So we're looking at machine learning and using AI to detect frag for aerial herbicide spraying and like super cool innovative stuff. So please register for that. I already know this is going to be such a cool talk. And thank you guys for watching. Thank you so much to our presenters. This was absolutely fantastic. I learned so much, and it was so good to hear an update on how your guys' work has been going. I guess if you wanted to, sorry, I should have put your guys' email up here so they could contact you. But I can put in an email with the recording if you guys are okay with that. So if you have any questions for Wes or Kaira,